So today I'm going to talk about an exploration of genetic resources of wild coffee species for the future of coffee breeding. So this project is in collaboration between Universidad de los Andes here in Colombia and the Institute of Research for the Development in France and the Université de Montpellier, also in France. So we are studying coffee because it is the second most consumed beverage around the world, with a production of 170 million bags per year. We have two species that are dominating the market: Coffea arabica, known as the Arabica coffee and the preferred one, and Coffea canephora, known as Robusta coffee. It's important to say that Coffea arabica accounts for 70% of the world production, and it is preferred because of less bitter taste that is a consequence of its lower caffeine content in comparison to Robusta coffee. However, Coffea arabica is adapted to highlands and is a more sensitive crop to changes in temperature and humidity than the Coffea canephora crops that are more resilient. Both coffee species are produced in this region between the tropics known as the coffee belt, with Brazil, Colombia, and Vietnam being the top three producers of coffee around the world. However, 50% of this production area is at risk due to climate change. So, because of that, now we have a question, and is if there's any natural source of resilience that we can use to improve those commercial crops. And we have in our knowledge that there are some wild coffee species that offer a wide range of valuable traits, such as adaptation to drought, as is shown here with this coffee plant growing in the middle of the sand. There's also some species that are resistant to the coffee leaf rust, that is an important disease for coffee, and could be more spread with some climate change scenarios. And we also have some variation in the ripening time, from days to more than a year in some wild species. The Coffea genus is composed of 130 species. That's the number we know now. It could be more. And the majority of those species are critically endangered, so it's important to understand them and their diversity. They are classified, or previously classified, into genus. The Coffea genus, which has the exposed reproductive organs, and the Silanthus genus, that has a hidden reproductive organs, being more on the aptogamous plant, but the Coffea species are more allogamous plants. Those coffee species show variation in the leaf shape and size, in the fruit shape and size, and also in the seed size. And those species are distributed in Asia, Africa, and the northern part of Australia. We have the Exylanthus species that are widely distributed, so we have some clusters from Asia and Africa. And within the Coffea species, like the originally Coffea genus species, we have 66 species that are endemic to Madagascar and Mascarini Islands, as are shown there in red. We have some species that are adapted to high altitudes in the central and east species in continental Africa, such as Ethiopia, and that shown here. We have another species that are adapted to low altitude environments in both sides of Africa, here in the west and central part of the continent, as well as in the eastern part of the continent. So based on all that information, we wanted to know how can we use genomics to study the wild coffee species, and we decided to sequence a total of 82 species using Illumina short reads, all of those species belonging to the different geographic groups that I showed you before. And as you can see here, there's still a lack of information about a lot of species. And we added some data from databases, but there was not a lot of information publicly available. As a result, we got over almost 7 million SNP markers that help us differentiate different groups.
So we had first the differentiation of the X Celanthus genus, here shown in yellow. We had the species from continental Africa clustered in 3 separate clusters. First, we have the low altitude species from East Africa, the high altitude species from Central and East Africa, and all the low altitude species from West and Central Africa here. Talking about the species from the Indian Oceanic Islands, we also had another cluster that is composed of species from Mascarenes and Madagascar. Those species were previously classified into some botanical series shown here. Each circle corresponds to one different species. However, when we analyze the structure or the differentiation between those species, using over 3 million markers, we notice that there's not a grouping that corresponds to the same botanical series that were classified according to some phenotypic traits. So we decided to assign a new classification. So here we have the Mascarene group that was previously reported, the Coffea group, and another 7 groups that need a deeper study and research to know if those genetic variants are related to any adaptation to specific environments in Madagascar. For now, we can conclude that wild coffee species evolution and adaptation could be studied through genomic variants. And we then wanted to know if complete genomes show the same variation that we were seeing when we used the Illumina data. So we decided to sequence 13 species using long read technologies, some with nanopore, some with papaya, well. And those species belong to the different phylogeographic groups that I previously described, as well as to outgroups from the Rubiaceae family. As a result, here we have a great variation in genome size. So we have first the Bungaria and Crossia genomes that are small genomes, and also the Coffea humboldtiana genome that is around 400 megabases in comparison to Coffea heterocalyx that has a genome of more than 600 megabases. This genome size, interestingly, is not correlated with the gene content. So the amount of genes within each genome is very stable, it's almost the same number. However, there's a high correlation between this genome size and the amount of transposable elements within the genomes. Well, it's supposed to be a gray thing there, but it's okay. Those transposable elements are mostly long terminal repeat retrotransposons, shown here in blue. And those elements are classified into 2 large superfamilies, Coffea and Gypsy, with a lot of different lineages that have diversified within the different phylogeographic groups and the species. At something important here, we found a great correlation between the amount of decadal transposable elements and the variation in the genome size, and also if we look into the data lineages. So, as a conclusion for this part, the size changes in wild coffee species are related to the variation of transposable elements, but there's also a need of more understanding of those patterns of insertion of those elements and their proximity of genes and more information. Finally, as we had all these genomes, we decided to reconstruct the first super pangenome of the Coffea genus, and we found that the genome structure is very conserved among genomes, so we have large synthetic blocks in both sides of each chromosome. We found that there are between 40 and 70% of genes that belong to the core genome and are shared between all the species we were comparing. And also, the pangenomics analysis revealed a large accessory genome within those Coffea species. This accessory genome is important because it can include genes that are related to adaptation and some quality traits. One of the important quality traits is the caffeine content. And it's important because there's a market of over 18 million US dollars per year of decaf coffee. So it's important to know if there's a natural source of decaf coffee. First, here we have that the caffeine content is a paraphyletic trait because we have species with and without caffeine and the different phylogeographic groups that we previously identified. The caffeine metabolism requires, or was reported to require, three N-methyltransferase genes, XMT, MXMT, and DXMT. We can talk about them later in the break. However, the pattern of presence-absence variation is more complex than saying just we need the three genes or we don't need them. First of all, we have the ancestral gene in chromosome 2 that is present in all the species, including the Rubiaceae outgroups. We have three NMT orthologs that are present in Coffea species but are absent in Rubiaceae species, but they are not functionally characterized, so we don't know what's the role in the caffeine metabolic pathway. We know that the absence of all the genes results in caffeine absence, as is shown here for Coffea pseudosanguevarii. We know that the absence of the last gene, the DXMT or the caffeine synthase gene, results also in caffeine absence that was previously reported for in Coffea humboldtiana. And we found here that the absence of the second gene, the MXMT, is compensated by other genes because the species that lack this gene can have caffeine within the plant. So now the question is, how many genes do we need to produce caffeine? And what's happening to compensate the absence of this MXMT gene? So as a conclusion, alternative caffeine metabolic pathways are potentially used in wild coffee species, and it's important to know them if we need or we want to produce some naturally decaf coffee. Finally, it was just an example of the potential of exploring coffee, the wild species of coffee, and how can we use all this information for breeding, for understanding diversity, for conservation purposes. So if you want to join us, collaborate with us, we created, or actually from out there, created this wild coffee database where you can have the information about each coffee species, the publications, some links to genomes, to data sets, the distribution of each species, and also some pictures, pretty pictures about the leaves, flowers, fruits, a lot of information. Well, finally, I would like to thank all the funding agencies and the British Coffea Consortium because we need a lot of hands to do this work. And thank you all for being here.
Thank you, Laura, from University of Montpellier, and yourself. Sorry, it was because the red background, I don't know. So we now have time for questions.
I have a couple of questions. Thank you for bringing us. I would like to know more about the coffee roast. If you guys are working on that line, identifying either resistant genes or varieties or species that may have like a differential resistance or immunity to the coffee roast.
I'm not directly doing that, but there are some of those species that have resistant genes. So one future work could be related to looking into the reported resistant genes and new resistant genes and see what happened. But I'm not working on that, so I don't know.
Thank you. And my other question, just very quick, just wondering what tool do you use to annotate the transposable elements in these genes?
Okay, that's a good question. So in our group, I didn't say that, but the majority of the work was performed using the tools that we developed inside our research group. That it's called NGSEP, the entire platform. So for transposable elements, we use the NGSEP transposon finder. That is a quick tool for that. But we also perform detailed work of annotation using EDTA, using the database of Impactor to annotate mainly LTR retro elements. Also use mask area. a lot of problems there.
Thanks.
Any other question?